Hello there, micro students. Just wanted to do a quick video about a part of the mixed unknown project that we're going to be starting soon, and that is called the dichotomous key. And so I'll talk about this more in lab this coming week, but I also wanted to make a video that you could use as a reference moving forward as well. So to understand how the unknown project is going to go, it's called a mixed unknown, and that's because two bacteria are going to be mixed together into a single tube. So each student is going to get a tube that has these two bacteria. And so what you see on this slide is a list of gram-positive possible unknowns, and as well as gram-negative possible unknowns. And so you're going to have one from each of these two lists. So one from this list and one from this list. And again, they're going to be mixed together into a single tube. Now, we just finished the labs that were dedicated to the gram-positive bacteria. And so this is a good time to talk about the dichotomous key that you can use to help you identify your gram-positive unknown. So that's what I want to focus on mostly in this video. So as you look at this list, there are 9 possible gram-positive bacteria, as well as 8 possible gram-negative. But again, the goal will be to focus on these gram-positive bacteria first, and that will be the focus of this video. So if you look, these are not just any random gram-positive bacteria, but they cover the bacteria that we studied during the 2 labs that we covered focused on gram-positive bacteria. So these first 4, Staph aureus, Staph epidermidis, Micrococcus luteus, and Micrococcus roseus, were the 4 bacteria that we saw when we studied the Staphylococcus lab. So if you recall, for that lab, each student chose an unknown bacteria that was identified just by a different color dot. And by the end of the Staphylococcal lab, you should have identified that color dot, that unknown bacteria, as one of these 4 Staphylococcal bacteria. And then we did something similar for the Streptococcus group. So these were the 5 strep bacteria that were with the focus of the strep lab. And so the unknown gram-positive bacteria that you're going to get in your unknown project is going to be 1 bacteria from this list. So again, not any random gram-positive bacteria, but one that we've been working with in the 2 labs that we previously have covered. Now, there was a question that we talked about during these labs, and it is now stated here. It says, what is the biochemical test that differentiates staph from strep? And so I don't know if you remember this, but there was only one test that we did in both of these labs, and all staph were positive for this test, and all strep were negative. Another hint is what we were looking for when we performed this test, and that was the formation of bubbles. So if that's not enough information, I'll just tell you what that test is, and it's called the catalase test. So as you might guess based on the name of this is a test, a biochemical test that is asking and testing for the presence of the catalase enzyme. And so as I mentioned, all staph have this enzyme catalase, so they are positive, and all strep do not have this enzyme, so they are catalase negative. So this can be seen in what we call these biochemical test tables. And so this was the table that we used for staph and Micrococcus as well. So across the top, these are the 4 possible gram-positive staphylococcal organisms you could have. But then on the bottom, these are the gram-positive streptococcal organism. So again, every student is going to have one of the bacteria, either these 4 or these 5. So it's a total of 9 possible gram-positive bacteria, either staph or strep. And based on what we just said, the catalase test is going to be really critical in determining, is my gram-positive a catalase-positive staphylococcal organism, or is it a catalase-negative streptococcal organism? And so this is a really helpful thing to think about, and this also helps us generate this this tool called a dichotomous key. So that's what I want to talk about on the next slide, is how do we take the information from a biochemical table and convert it to a dichotomous key. So it's important to remember that all staph are catalase positive and all strep are catalase negative. That information is going to be really important in the first part of how we generate a dichotomous key. Okay, so that's where we're heading next. So what I have at the top here is the list of all 9 potential gram-positive bacteria. So you can count them. I've abbreviated them a little bit, but hopefully you can decipher that. So Staphylococcus aureus, Staphylococcus epidermidis, Micrococcus luteus, Micrococcus roseus, etc. And then these are all short for Streptococcus. So as we just mentioned, there's one important biochemical test that will be able to answer the question, is my gram-positive one of the staph group or one of the strep group? And of course, what I'm talking about is the catalase test. So this is the beginning of a dichotomous key. So what this is showing is that when you start this project, what you will know is that you have one of these 9 gram-positive bacteria. You won't know if you have a staph or a strep organism, but you have one of those 9. But if you do a catalase test, or not if, when you do a catalase test, you are only going to get one of 2 results. You either are going to see bubbles, which is positive, or you're not going to see bubbles, which is negative. And so those are the only 2 possible results that you'll get. And when you get those results, you also know which of these bacteria yields each of those results. So all the staph are positive and all the strep are negative. So without being able to see the future, what we can say with certainty is if you are given one of these 9, when you do a catalase test, you will either see bubbles, positive, won't see bubbles, negative. And once you have either of those results, you can narrow down what the potential list is going to be. So if you see bubbles and you conclude it's positive, you have one of these 4, and you can't have one of those 5 because those are always negative. But by contrast, if you don't see bubbles, then the bacteria are negative for catalase, then you have one of these 5. And so with each test that you perform, you should narrow down and reduce what your potential bacteria could be. Now, of course, if you got a positive catalase test result and narrowed it down to these 4, there are additional tests that you can do that will allow you to narrow down this group of 4 even further. So a question that you can ask is, what would be, if I got this positive catalase test, what would I do next? And so that's what this branch is supposed to show. And what you would do is use a biochemical test table like this to try to come up with what would I do next? And there isn't one correct answer. There are multiple answers. So glucose fermentation is a test we did. Coagulase is a test we did. Salt tolerance is a test we did. So there's not one correct answer. I'll show you one more example just to hopefully get the idea kind of seated in your head and you can take it. And if not, you can ask questions in lab. And that is just looking at the glucose fermentation test. So that's this row right here. So if you look, of these 4, 2 are positive and 2 are negative. This is for the glucose test. So if you did a catalase test and it's positive, you know you have one of these 4. If you do a glucose test and it's positive, you now know it's one of these 2, one of the 2 staph organisms. So Staph aureus is positive and Staph epidermidis is positive. So now you've narrowed it down to those 2. But if you did a glucose test and it's negative, there are only 2 that are negative, and that's Micrococcus luteus and Micrococcus roseus. And what this doesn't show is there would be another test that you would propose under this one. So you'd have to ask the question, what test could I do to identify between these 2, Staph aureus and Staph epi? But then you'd also have a dichotomous key branching that comes from the 2 micrococcal species. So what test could I do to differentiate those? And then, of course, we haven't even talked about it, but there is a biochemical table that helps you identify the strep group. And so that too could be get this pull up again. Get this pull up. So this is showing what test could you do to help identify within this group. And so again, the goal of a dichotomous key is basically generating a roadmap so that regardless of which of these organisms you have, you'll have a path that you can follow and it'll take you down to an identification of each of those. So this should be enough to get you started. But I just wanted to give you kind of a summary of why this is important. So as part of your mixed unknown project, you're going to have to submit two dichotomous keys as part of what you'll be graded on. And so you're going to have to generate these anyways. These dichotomous keys are really helpful in planning and carrying out the experiments and what tests in what order that you will do to identify your unknown. And so it's really helpful to make sure that your dichotomous key is correct and complete before you start using it for this project. And so what I offer to students is there's an assignment link on Canvas, but it's currently set for 0 points. And that just provides you the opportunity to submit a dichotomous key before it's actually due with the mixed unknown. And I will look at it, review it, and make comments. Most importantly, if there are any mistakes, I'll point them out to you. And so you can correct them and make sure you're using a correct dichotomous key. Or if they are 100% correct, then you'll know that going in. And so that will allow you to ensure that you have a correct key starting the project. And you basically can be sure that when you submit them as part of the project, that they are complete and they are correct and that you'll earn all the points for those dichotomous keys. So I just want you to be aware of that possibility and that option and make sure that you're using it if you are interested in doing so. And like I said, you're going to have to turn them in as part of your project anyways, but if you want to turn them in or generate them and turn them in at that time, that's totally up to you. But just want to make you aware of this opportunity. So we'll talk more about this upcoming week in lab, but just wanted to kind of get you a little bit started on this in case you want to think about it and generate questions before our labs on Monday and Tuesday of this week.